So it is with great pleasure that I introduce the Squibb Professor of Molecular Biology and Chair of that department, Dr. Bonnie Bassler, who's coming from Princeton University, to give the second of 3 Prather lectures, which, as you heard, is a joint endeavor by the OEB and MCB departments. And the nomination alternates between those departments each year. But this year, it was an easy decision because Bonnie's work spans the breadth of both of those departments and beyond. For her work starts by elucidating the protein structures of the molecules that make up the language and communication between microbes, all the way up to their collective behavior and their role in the complex microbial ecosystems. The other reason we are so excited to have Bonnie here is because of her passion for and perfection of science communication, and I'm sure you'll get to experience some of that tonight. So to give you a very brief background on Bonnie, she received her Bachelor's of Science in Biochemistry from UC Davis and then went on to earn her PhD at Johns Hopkins. She joined the Department of Molecular Biology at Princeton in 1994 and has been there ever since. So Bonnie is best known for making key insights into the ways that bacteria talk, not only to each other, but to other species. And she has decoded their secret language. This work has clear applications in medicine, from offering novel ways in which we can interfere with bacterial pathogens to modulating our own microbiomes. For this work, Bonnie has received numerous accolades, too many to list, but here I'll just highlight a few. She's been awarded a MacArthur Fellowship and elected into the National Academy of Sciences, the European Molecular Biology Organization, the American Academy of Microbiology, the American Academy of Arts and Sciences, the Royal Society, the American Philosophical Society, the National Academy of Medicine. I could go on and on. She's also an investigator of the Howard Hughes Medical Institute and recently completed a 6-year stint on the National Science Board, a position to which she was appointed by President Obama. In addition to all of this, Bonnie is also known as the world's best, and perhaps only, bacteria whisperer. So with that, please help me welcome Dr. Bonnie Bassler.
Thanks, Hopi, I think. I am absolutely delighted to be here. Thank you for having me, and to both departments. It's such a treat and an honor to get to give this talk and to actually give this series of talks, and I just thank you really deeply from my heart for just a wonderful, welcoming several days of sort of living in my imagination through the work that's being done here. So thanks. Okay, and so now I have to actually come through on that, I guess, and so I will try my best. And so what I want to just say is that I have one goal for this talk, and that's to try to convince you that bacteria can talk to each other. And if I manage to do that, sort of secondary goals is I want to try to convince you that they speak multiple languages, and then I want to try to convince you that they work as groups, and that learning about this actually will give us new insights into how we can either get good bacteria to do stuff better or stop harmful bacteria from doing things that are hurtful to higher organisms. So that's where we're going. And so I want to make sure that, and I'm going to give sort of a historical timeline because this work actually started at Harvard, not by me. Anyway, so that's what we're going to do tonight, and so I want to just make sure that we all sort of start on the same page, and so I want to just introduce you to the critters that I love the most, which are the bacteria. And so bacteria are the oldest organisms on Earth. They've been on Earth for 4 billion years, and they're the most diverse organisms on Earth. But they all pretty much look, if you looked under a microscope, you need a microscope to see one, they look pretty much like this cartoon, which would be like an E. coli that's living in your gut. So they're single-celled, and they're covered with a membrane, and that's like their skin that keeps the outside out and the inside in. And then inside of this membrane are all these biomolecules that are roiling around that keep the organism alive, but then in blue, in this dark blue, they have one piece of DNA. So they only have one chromosome. And so they have a few thousand genes. So for comparison, you have 20,000 or 30,000 genes, and bacteria has a couple thousand. So they're really stripped down, not very much genetic material, but that DNA has the information that gives them all their physical traits and it also gives them their behavior somehow. So if you look at bacteria under microscopes, and people have done this for a few hundred years, and you watch what their life is like, it's actually kind of boring. They eat nutrients from the environment, they grow to twice their size, they double their components, and they divide down the middle, and one cell becomes 2, becomes 4, and so on and so on. And so it's kind of a mundane, boring life. And so for the past almost 150 years that people have been studying bacteria, because they live this seemingly boring life, scientists have thought they were these asocial recluses. And what I mean by that is when the cell divides in 2, it has no knowledge, if you will, of its sister cell. And I want to try to prove to you that that can't be true, because that they can communicate and that this gives them a lot of power. But before I get to that, I want to really try to trick you into listening to this talk. I want to tell you something about your interactions with these bacteria, which you've never seen with your eyes. So now what I will give you is the Bonnie Bassler view of the world. So here is my idea of a human being. And so what I've done is I've taken a metric and I've made all of these circles are supposed to represent some of the cells in the body. And we know relatively how many cells there are in the human body. There's a few trillion cells. So that's how many cells it takes to give you your physical form and then all of your traits. So if you look at a human being starting from a couple months after it's born, it turns out there are 10 times more bacterial cells than human cells. So if you want to use this kind of a metric, you're actually 10% human, you're 90% bacterial. Okay, but of course you guys are at a science talk, you know it's the 21st century, it's all about the genome, it's about the DNA, and of course we know the human genome, right? So that's been decoded in Hopi's words. And so I've made my man out of all the A's, G's, T's, and C's that make up the famous double helix. And so I already told you it's about 20 or 30,000 genes to make up a human being. In fact, you have 100 times more bacterial genes than human genes in you and on you at any moment of your life. And so you can pick either of these metrics. At best, you're 10% human. Really, you're only 1%. Because the other 99% of that information comes from these bacteria. And these bacteria are not passive writers. The genes that these bacteria encode give you all kinds of traits that your own human genome doesn't encode. And so what we now get is that these bacteria are your 24/7 partners, and they are actually giving you life because they give you access to all kinds of things and functions that your body can't do with its own genome. And so now what we now get is that there would be no life on Earth without the bacteria, because every higher organism, every animal, every plant, every human lives in a consortium with this microbiome that is keeping it alive by supplying it all of these other features. So that is your life. But now I want to go back to what you've probably heard more about. So we're only now learning about this microbiome. But of course, what bacteria have been known for traditionally is for causing disease. And so here what you're just looking at are a bunch of pictures of bacteria that have been taken under the microscope. So historically, bacteria have been known because they can make plants, animals, and humans sick, or they can kill us. So there's all these bacteria in the environment that have no business being in or on us. And if they are, they can bring you down. And then what I've just alluded to is that we're starting to understand that there's this magical conglomerate of bacteria that live with us all the time and give us healthy functions. And so we don't know everything this microbiome does, but some things we do know. So for example, when you eat a salad, when you eat plant foods, you don't have any enzymes to break down plant foods and give you access to those calories. Bacteria do that for you. They make lots of our vitamins and micronutrients, and they also educate your immune system and tell it to keep harmful microbes out and let good microbes in. And so these are just some of the traits that we know about. And there's going to be a lot more as this field matures. But the point for tonight is whether you want to think about all the harmful things that bacteria do, or you want to think about all the beneficial things that bacteria do, the question that my lab wants to ask is, how can they do it at all? They're so tiny, right? You need a microscope to see these. So how can it be that these itty-bitty ancient organisms can either kill us or keep us alive? And that's the question that is at the heart of our studies. And I want to try to show you that they talk to each other and they work in groups and they carry out tasks as collectives that would never be successful if they acted as individuals because they're too small to have an impact. So that's what my lab works on. And so again, I already told you that this all started here at Harvard by a former scientist who's unfortunately passed away from this department. His name was Woody Hastings. And it all started when he noticed that a very beautiful marine bacterium, which is named Vibrio fischeri, had the special property of bioluminescence. So it made light, like fireflies make light, except the light was blue. And he was very interested in studying how that bacterium made light. And what he knew about that bacterium is that it lived in a beautiful one-to-one symbiosis with this animal, which is called the Hawaiian bobtail squid. And so I'm going to tell you about this symbiosis first. And first I'm going to tell you about it from the squid's perspective, and then I'm going to tell you about it from the bacterium's perspective. So it's this one-to-one symbiosis. The squid needs the light from the bacteria, and then the bacteria is willing to give the light because the squid feeds it. So first let me show you something about this squid. So this squid, the Hawaiian bobtail squid, it's about this big, full grown, and it lives off the coast of Hawaii in just sort of shallow, knee-deep water. And it's nocturnal. And so what we've done here is we put it in an aquarium and the lights are on. So during the day, this squid sleeps. And so what you can see is that the light is on, right? And so it looks kind of spastic, right? Because it's not supposed to be out during the day. Because if it was out during the day, it could be eaten by a predator. So its trick that the squid does is when the sun comes up every morning, it goes out at night to hunt. And when the sun comes up, it buries itself in the sand to escape from predators. And so now we've given it some sediments. The light is on, thinks it's morning. And it's got to hide from predators. You probably just saw it camouflage itself. It's thorough. You only get eaten by a predator once. Right, so now she's going to finish burying herself, right, and then in a second it'll take its tentacles back in. And if I showed you this aquarium, of course you wouldn't know that there's a squid in there. There's actually about 10 of them in there. Okay, so fantastic strategy. In the daytime, the way the squid manages to not be killed by a predator is to bury itself in the sand and be invisible. But it can't be there 24/7. As I told you, it's got to come out to hunt. And so this is where the problem arises and where the bacterium comes into play. So I told you it just lives in this shallow, knee-deep water. So when it comes out at night, when it emerges, right, to go hunting, since it's just in this couple feet of water, the light from the starlight or the moonlight can penetrate the depth of the water that the squid lives in. And so if it was swimming around, that light would shine onto the squid, and the squid would cast a shadow onto the sea floor, and then predators could see the shadow, calculate its trajectory, and eat it. So what the squid has done is to develop this beautiful symbiosis with this Vibrio fischeri, glow-in-the-dark bacterium, to counter-illuminate itself. And so the way that it works, so now I've taken the squid and it's been turned on its back, and I hope what you can see is these glowing orbs. So what this squid has, it has a specialized light organ under the mantle or the body of the squid. And in this light organ, Vibrio fischeri is housed there at something like 10 to the 12th cells per milliliter. So it's like paste, it's like toothpaste of bacteria. They're in there, and I told you these bacteria are bioluminescent. So they are making light. And what the squid does when it comes out to hunt is it has detectors on its back. So it can sense how much starlight or moonlight hits its back. And then it opens and closes a shutter. It's the squid's ink sac under this light organ. So the amount of light that hits its back exactly matches how much light comes out of the bottom that's made by the bacteria. So when it swims around, it doesn't make a shadow. Right? So this is like the stealth bomber of the ocean. Right? It cloaks itself in this invisible device. And now it's got a fantastic strategy in the day and a fantastic strategy at night, right? And so that's how it works, but now I want to tell you about it from the bacterium's point of view. So it turns out the bacterium only makes light at night. So how can this bacterium know whether it's day or night? And this is the part where it matters that the bacteria talk to each other. So what happens is every morning when the squid buries itself in the sand, it's got a pump that's attached to its circadian rhythm. So it has this thick culture of bacteria. They're making light. It squirts out 95% of the bacteria. So now they're dilute, right? And they actually stop making light. And that's good because the squid can't feed them indefinitely. And so then as the day goes by, the bacteria grow to this high cell number, and then at night the light comes on again, right? So they only turn the light on at night, and the squid is making sure they're only at high number at night. So how does that happen? And that's where this communication occurs. So now we're going to switch for the rest of this talk to the bacteria. Enough of these lower organisms. Okay, so here's my cartoon of a bacterium. So in the morning, right, it's at low cell density, right? The squid has pumped out most of the bacteria. So even though it's in this little light organ, the world is big and bacteria are small. So in the morning, the bacteria are at low cell density, and so they're not making light. But what they do is that they make and release small molecules that I have depicted by these red triangles that you can think of like hormones or pheromones, and we call them autoinducers. And so what happens is, since the cells are in dilute number in the squid light organ, they make and release these autoinducers, they diffuse away, and the bacteria can't detect them. And when they can't detect them, that says don't make light. But then as the bacteria grow and divide, like I told you on my first slide, since all of the bacteria are making a share of this hormone, this autoinducer molecule, the concentration of that autoinducer outside of the cell increases with cell number. And when that molecule hits a particular threshold level, the bacteria detect it. And what they infer from that detection event is that they must have neighbors around. And so in unison, all of the bacteria turn on light together. And so, in fact, they don't know how many cells are around. They're measuring the concentration of this chemical, and they use it as a proxy for cell number. But what I'm trying to tell you is that this molecule is letting them talk to each other, if you will, to know that one another is there. Right? So this is how it works, and we know we can take these bacteria out of the squid, and we know this is true because we can just collect up that molecule, and if we squirt it onto these dilute cells, they will turn on light. Right? So they have no idea there's other cells around. The chemical tells them the cell number. Okay? So that is how that works, and then you understand how the squid makes it so it's like this in the morning and this at night. Yes? I usually get to the fourth slide. Go ahead. Yeah.
Why is it producing that light? Just because others are around?
Oh, no, so yeah, exactly. So this is a philosophical question because most things in the ocean are bioluminescent. But remember, the squid, so they are growing to 10 to the 12th or 10 to the 13th per mil in this light organ. And when they get out, they're still alive. But the ocean's like a desert. So there's no way for them to grow. So presumably, evolution has selected that there's a benefit from making this light, which is you get to live the good life. You know, there are all these goodies. Sorry, I should have said, there's all these goodies in this light organ that lets them make more progeny. And that is presumably the selection for them to spend the energy to make the light. Other questions? I see how this is going to go. Anyway, I'm glad to answer questions. Okay. All right. Okay, so that's the phenomenon. And then, of course, we wanted to understand how does that actually work from the bacterium's point of view. So we could bring the tools of molecular biology and genetics to the table and actually ask, what are the components involved in making this? And so now this is supposed to be my bacterial cell a little bigger. So what we found was that there was a protein, an enzyme, that makes that hormone molecule. And that can just slip across the membrane, right? So this enzyme synthesizes this molecule, it slips across the membrane, and so as the cells grow, the concentration of that molecule outside the cells increases. And then there was a partner protein, which is a receptor, and this sits on the bacterial surface. And so the receptor connects the outside world to the inside world because it spans the membrane. And so this is very much like the receptors, your hormone receptors that sit on your cell surfaces that tell your cells to detect things that are happening in your body and then behave, right? So what happens is this autoinducer increases, and then literally like a lock and key, it slips into the, I've actually never given 3 lectures in a row. We don't actually work at Princeton. Anyway, so I actually drink coffee and yell at people mostly. And so anyway, okay, so this molecule, you're not actually taping this. So this molecule goes in here, and what happens is when the molecule, like a key in a door, goes in, it unlocks, it changes the protein's conformation, and then information comes into the cells to tell the bacteria to turn on the genes that make the enzymes that make light. So it's a really simple circuit. The more bacteria there are, the more the hormone there is. At a particular amount, all the bacteria undergo that, and they turn on light in unison. And so that is how it works in Vibrio fischeri, and the reason that Hopi invited me here is because what we realized after sort of figuring these things out is that this is not some crazy anomaly of the ocean. So once we got into this, we and lots of other scientists started to look for these little circuits. And at last count, there are hundreds and hundreds of species of bacteria that are kind of built the way Vibrio fischeri is, in that they all have an enzyme that makes an autoinducer molecule. There's always a partner protein. And then together, these complexes turn on all kinds of genes that bacteria want to express when they're in communities, but not when they're alone. And so now we have a fancy name for this process. We call it quorum sensing. They vote with these chemical votes, the vote gets counted, and then they behave in this sort of democratic decision-making process, and they do jobs. And why this is especially important, and Hope already told you about this, is because in many, many cases, these are in clinically relevant bacteria. And so what they're turning on is biofilm formation, which is how bacteria sit on surfaces, and the group-wide production of virulence factors. So what we know is that if we make mutants that can't talk or can't hear, they're completely avirulent, right? So the idea from the pathogen, now I'm talking about pathogen's point of view, is that the sort of dumbest thing a bacterium could do is if a couple bacteria got in and they started dribbling out some toxins, it's not going to have an effect. And those are red flags for the immune system. So the better idea from the bacterium's point of view is to wait, to count yourselves and recognize when you have enough bacteria, if they all launch these virulence cascades together, they're going to be able to overcome the host. So now we have many cases that we know, many cases where quorum sensing controls virulence, and if the bacteria don't have quorum sensing, they're avirulent. Okay, so this seems to be the norm in the bacterial world, whether you're a pathogen or not a pathogen, that being able to carry out collective behaviors, right, and make what I would call public goods, where the bacteria give stuff away. They never get their own product back. It's only when they do it together that they get the benefit of one another's work. That's quorum sensing. So that's how it works. And then, of course, these molecules are actually chemicals. And so we and other scientists started to figure out what are the actual chemical words that these bacteria talk with. So, of course, the first one, and this was discovered here, that was discovered is this one from Vibrio fischeri, because that's the bacterium we've known about the longest. So in my cartoons, I'm using a triangle, but this is the actual chemical that is the word. And then we and others started to look in lots of other bacteria. And so what you're looking at here are just a smattering of different species of quorum sensing bacteria, and then the molecule that each one of them uses as its word. And what you're supposed to see, which I know is that the left-hand part of all of these molecules are identical, and then the right-hand part, these are just carbons, is a little bit different in every single species. And these tiny differences confer exquisite species specificities to these languages. So these really do fit like keys in doors. So you know how all your keys look the same, but only one unlocks the door? That's how these work. So what I mean by that is if we take the Vibrio fischeri molecule and put it on Agrobacterium, nothing happens. And likewise, Vibrio fischeri is impervious to Agrobacterium's molecule. So what we understand is that these molecules allow intra-species communication. This is how I count my siblings. These are private, secret conversations that bacteria carry out with their kin to decide to do something. So that's the first part of quorum sensing. And once we sort of got that far in our studies, my lab was really interested. Like, it's really great if you're Vibrio fischeri and you live in this pristine light organ and there's no other bacterium there. But that actually isn't how most bacteria live. They kind of live in the Wild West with lots of other species of bacteria. So this is a bacterial biofilm that I've said that word before. This is human skin. So this is what the skin of your elbow would look like if you looked at it under a microscope. It's covered in a bacterial biofilm. And the point, that's not actually the point. The point is that each one of these different shapes and sizes is a different species. And so if quorum sensing is so species specific, like I just told you, the question that we next wanted to ask is how do these bacteria take a census of the community when there's all these other species present? So we wondered, couldn't quorum sensing be more sophisticated than what was originally learned in Vibrio fischeri? And so what we decided to do to explore that question was to move to a cousin of Vibrio fischeri, and that's a bacterium named Vibrio harvii. So you're going to ask me the same question. Vibrio harvii is a bioluminescent marine bacterium, but it lives free-living in the ocean, and it makes light in the free-living state. Right? And so what we thought about that bacterium is this is great because it's got to encounter all these other species of bacteria. We knew that it only turned on light at high cell number, so we knew it had quorum sensing, and so we thought maybe we should try to study this bacterium's circuit to understand how does a free-living bacteria count. And so the reason that I'm showing you this slide, I should tell you what this is. This is a flask. These are Petri plates. This is what the graduate students in my lab do. Right, and all we did, so I'm showing this because I actually want you to understand the experiments that we do. All we did to take this picture is turn the lights off in the room. We actually took the picture from the light the bacteria are making. So we just see this when we turn the lights off. And so what was so great, and this was Woody Hastings' first insight, right, is that bacteria are invisible. Right? And so you can't tell if they're doing something alone or something together. What was so fantastic about bioluminescence is you could just see it with your eyes. And we could see that they turned the light on together. Right? So this light gave us this visible way of watching bacteria behave. And then in my lab, we're geneticists. And so what we could do is just randomly make mutations, put the bacteria on Petri plates, turn the lights off in the room, and look for guys that didn't make light. I mean, it was super easy. Like, they were dark when they should be bright, or bright when they should be dark. And that's how we found all the components I told you about and the ones I'm going to tell you about. So it was really a simple trick that the bacteria gave us by having this bioluminescence. Okay, so now we're going to do that in Vibrio harvii. We're going to ask what are the genes and proteins and molecules involved. And so when we did that experiment, what we found was indeed there was an enzyme that made an autoinducer. I actually showed it to you a couple slides ago, and it had a partner receptor protein that detected it and sends information into the cell. But what was interesting is that when we knocked out these components, this bacterium still made light at high, and only at high cell number. So there had to be a second system. And so we found those genes by doing what I'm telling you. And so we found a second enzyme that makes a second molecule, and it also has a partner receptor protein. And then all of that information comes into the cells to tell the bacteria to turn on light. And hundreds of other genes. We are only measuring light in our experiments, but quorum sensing is about much more than light. It's about going from being alone to being as part of a group. So hundreds of genes have to turn on and off for bacteria to make that transition, and we're measuring one of them, which is light. Okay, so what we realized was that there were two systems. And so our next question is, why did evolution make this so? If these molecules don't encode different pieces of information, having two systems shouldn't be better than having one. And so we wanted to try to understand what the roles of each of these molecules were. And I already told you about this one. It's for intra-species communication. So what we did was we started to just scan any bacterium we could get our hands on and ask, does it make this molecule? And sure enough, unlike this one, which is only in Vibrio harvii, every bacterium we tested made this molecule. So we purified that molecule from lots of different bacteria, like I showed you on the first slide, and said, what is it? And what we found is that no matter what bacterium we purified it from, it was exactly the same molecule. So this is what we call autoinducer 2, because it was the second one that we identified. And these are just carbons. It's a 5-carbon molecule decorated with all of these oxygens. And that part isn't important. What's important is that no matter what species across the bacterial kingdom we purified this molecule from, it was the same exact molecule. And so what that told us is that this can't be like that first language where each one's a little bit different. What we think this is sort of the trade language of bacteria. It's like the bacterial Esperanto, that they can talk across species because they have a common language, and that is this molecule. So we think this is sort of a universal interspecies, between species language. And so now what we think is that bacteria have to at least be bilingual. I will still have a job when I go back tomorrow, so there's probably more molecules to find. But what we think then is that bacteria have to have some way to know self and then some way to know other. And so I think that the computation that bacteria do is the following. I think when they're growing out in the environment, the first thing they do with these receptors is they're just detecting, are there quorum sensing molecules around? And then that tells them you're alone or you're in a group. And then they start to turn on and off genes down here. But then the more interesting and sophisticated computation that we recently showed they do is they actually measure the ratio of these molecules. And then they change what genes they turn on and off based on whether they and their kin are in the majority or somebody else is, right? So we think they can tell self from other, and that they actually measure how many of me, how many of the other guy, and then they behave appropriately based on whether they're collaborating or they're in fierce competition, for example, with the enemy. And so that's what we think quorum sensing is about right now. And then I've already told you that these systems, and I should tell you both of these systems, control virulence in all kinds of pathogenic bacteria. And so now that we've learned about this by doing sort of basic discovery science, the question is whether we can take what we've learned and actually, think about doing something useful, like I kind of think that was useful, but make an actual application that would have a good use. And so there's sort of two strategies. And one is that you could try to interfere with, like if you could shut down intra-species communication, maybe you could make a new kind of antibiotic. Like, could you make a medicine, for example, if you had a specific disease like Pseudomonas or staph, and you just want a medicine for that, maybe you could take advantage of this intra-species communication molecule. Lots of times, like if you're sick and your physician doesn't know what's wrong with you, you get treated with what's called a broad spectrum antibiotic that works across species. And so in that case, since this second autoinducer is generic, maybe we could interfere with that and we could make a broad spectrum a new kind of antibiotic. And so that's what I want to talk about for the rest of my talk, but I want to make an interlude about antibiotic resistance. So everybody in this room has heard that we have this global problem with antibiotic resistance, right? There aren't any more in the arsenal. And so what I want to do before talking about whether you could interfere with cell-cell communication, I want to actually talk about antibiotics and how antibiotics work and why there's this resistance problem. Because everybody's heard of that, but I thought you might want to know how that and why that happens. So here's some famous antibiotics. Antibiotics are just chemicals. So everybody is familiar with those names. So these are the actual structures of some very famous traditional antibiotics. And what antibiotics do is they stop bacteria from growing or they kill bacteria. So that is what an antibiotic is. And they all work by a couple of different mechanisms. So some antibiotics come into, this is supposed to be a bacteria, into a cell, and they stop it from making copies of its DNA. If you can't make DNA, you can't make the next cell. Some antibiotics, there's sort of three important components. There's DNA, then DNA gets turned to RNA, and RNA turns to protein. And so if you can't make any one of those, you can't grow and you can't make progeny. So antibiotics either stop DNA, RNA, or protein synthesis. Other antibiotics make holes in the wall, and so all your guts spill out, right? And so they punch holes. And so these are sort of the mechanisms that any antibiotic you've ever taken works by one of those mechanisms. And so what's resistance? So resistance is when bacteria get fortuitous mutations that make it so one of those four things don't happen. So for example, bacteria could get a mutation, right, so that they have pumps that pump out byproducts and waste products, and maybe that pumps out your antibiotic. So now you're resistant because the antibiotic doesn't stay in the cell. Likewise, bacteria can get mutations in these components, so the components work, but the antibiotic doesn't bind, right? So they're resistant. They have enzymes, bacteria, like we do, they have enzymes to do all kinds of jobs. Maybe some random enzyme chews up the antibiotic. Then the bacterium is resistant. They can also put extra components into their cell wall to make their cells, them not permeable. So resistance is simply mutations in these functions that make it so the antibiotic can't do its job and the bacteria can make more of itself, right? So that's resistance. It's just fortuitous mutations which happen whenever bacteria, whenever they replicate their DNA, make more themselves, fortuitous mutations happen, and occasionally it's in these components. So the way this resistance, so that's resistance, but the way the antibiotic resistance problem that we think about happens is like this. So in any population of bacteria, there are pre-existing mutants. There are just mutants in this population, and some guy in this population of bacteria will have fortuitously gotten a mutation that makes it resistant to an antibiotic. Right? So it's pre-existing in the population. So if you got an infection, these guys are in your gut or whatever, and then you supply an antibiotic, right? The antibiotic, so the blue is supposed to be the traditional antibiotic, will kill off most of the bacteria, right? But not this pre-existing mutant that is resistant. So if you kill off these guys, you give a lot more real estate for this guy to grow. It's resistant. And so what happens is that it and its offspring grow selectively. So now the whole population is resistant, right? And so this is why antibiotic resistance is so prevalent, is because when you put the antibiotic medicine on, you kill off anybody that's not resistant, and you give an amazing advantage for growth of these resistant mutants, and then they get around. And so that's how resistance arises. And so now what I want to try to argue is perhaps you could think about making what we're going to call antibiotics by a new means. Like if you could stop cells from talking to each other so they can't count and they can't launch these cascades, could that be a new way of making medicines? And might it have a longer shelf life before bacteria get resistant? So I've just told you in a traditional antibiotic, right, most of the non-resistant cells die, the resistant cells grow and divide. So what if you made an anti-quorum sensing therapy? So now you're going to have pre-existing mutants that have quorum sensing, and they're not. And if you made an anti-quorum sensing molecule, they're not affected. But you haven't actually killed anybody else. You've just shut down quorum sensing. So then these guys, the red guys, don't get a selective advantage. And so maybe if you could actually interfere with bacterial behavior instead of growth, maybe you could get a medicine that would have a longer shelf life before the bacteria acquired resistance, right, and actually made the medicine useless. And so we're very into that idea of this behavior modification and whether or not that could be a new way forward for antimicrobials. Okay, so so you get why that might be better. We don't know that, but it might be better than a traditional antibiotic. It might not make resistance happen quite as fast, right? And so then the question is, can you actually do it? Okay, so now I want to just show you our first steps towards doing that. So obviously you're not going to do this in Vibrio harvii or Vibrio fischeri because they're not pathogens, right? So we don't need to make an antibiotic for them. So what we turned to was a bacterium named Pseudomonas aeruginosa. And so this is a notorious pathogen. It just lives in the soil. You and I encounter it every day, and it's harmless. It's an opportunistic pathogen. So it's harmless unless you have the disease cystic fibrosis or you're immune compromised. So you probably know that people who have this disease, CF, they have a genetic mutation in their lungs, so they can't clear their lungs. So you and I breathe in all kinds of gunk, but we have mechanisms to keep our lungs sterile. People that have CF can't do that. And so they always have this polymicrobial infection. And what happens is typically, for reasons that we don't understand, was that when a kid is in his or her teens, they become permanently colonized with Pseudomonas aeruginosa, and that's what kills people that have CF. They die of a Pseudomonas infection. Pseudomonas is also takes advantage of people who are immune compromised, so people in hospitals, right? It's also a notorious biofilm former. So I already told you biofilms are how bacteria stick to surfaces. This is a really sticky bacteria. So in hospitals, people are sick, Pseudomonas is there, and so it's one of these sort of escape pathogens that's sort of running rampant in hospitals because it loves to stick to surfaces. And so it can be transferred around in the hospitals. It's the thing that, like, when you go in and you get a catheter, you get an intubation tube, and then you come out with a bacterial infection, it's that guy, right? So it's a real problem in hospitals because it's a biofilm former and it's it has virulence, it's a pathogen. Both of those things are controlled by quorum sensing. So just to remind you is that Pseudomonas uses this mechanism that I've been talking about. When it grows to high cell numbers, it makes a biofilm and it secretes like 40 or 50 pathogenesis toxin products, right? And that's what kills people in hospitals or people that have CF. So this is a good bacterium for us to test this out on because it's a relevant bacterium, right? And we know that quorum sensing controls the show. So what we wanted to do is to try to interfere with quorum sensing. So now I'm going to remind you, I actually showed you this before. This is the autoinducer. This is the molecule that Pseudomonas uses to talk with. It has two. It has one that has a 4-carbon tail and one that has a 12-carbon tail. So I'm just showing you one. So what we did was we made a synthetic molecule. This is the structure of it that looks kind of like the real molecule. So if you look at the left-hand part, only this sulfur for this oxygen is different. So the molecule looks kind of like the key, but it's got this big bump on the right-hand end. And so what happens is when this molecule goes into the receptor, it jams the receptor. It doesn't turn on quorum sensing, but it blocks the other real molecules, these, from getting in. So this is an antagonist, right? It antagonizes the natural receptor. So we made that by knowing what that molecule looks like. Okay, so now we have this antagonist, and I want to show you at least the proof of principle that this idea could work for shutting down virulence. So I told you that quorum sensing controls biofilm formation and virulence factor expression. So first about biofilms. So now you're looking at a Petri plate, and what we've done is we've just put a little dot of Pseudomonas aeruginosa in the middle of the Petri plate. And then overnight, the bacteria swarms out. It makes a biofilm on that surface, and it swarms out, and it makes this, to me, this beautiful pattern. So that is biofilm formation. If into this plate we put that inhibitor, that antagonist, what you can see is that the bacteria can't make the biofilm, right? So that was the first good news. It looks like it shuts down this swarming business across the plate. Okay, so that's step one of the infection. Step two is to secrete the toxins, the poisons that actually make human beings sick. So Pseudomonas, I drew my cartoons in green. That bacterium, if you looked at it under a microscope or if you look at it in a test tube, it's green. And that's because it makes a toxin that has a green color. It's called piocyanin, like for cyan. So here is just regular Pseudomonas grown to high cell number. Quorum sensing is happening. The whole culture turns green because it thinks it's a pathogen, and it secretes this green toxin. If we make a mutant that can't talk, can't hear, what you can see in this test tube, they're colorless. So that shows you that quorum sensing controls production of that green poison. So then if we take this bacterium, but we add our inhibitor, what you can see is that they're pale green. And so in fact, we can shut this down 90%. Right, so now I've shown you two things. On a Petri plate, they don't make a biofilm, and then in a test tube, they don't secrete this very important toxin. So that looked pretty good. And so the question is, can you actually do it in an animal? And so that's the final experiment that I'll show you. So we have an animal infection model that was actually provided. Where did he go? Provided to us by Fred Ausubel. There he is. He's sitting back there. Right, who's a Harvard scientist who developed this animal model for testing Pseudomonas virulence. And he gave us this model. And so what one does is one measures how many animals are alive over time. And so obviously, if we don't infect the animal with Pseudomonas, they're going to live just fine. If we give them a Pseudomonas infection, they all die within a day. Right, so that shows you Pseudomonas is a pathogen. That's not going to be a surprise to you. But if we do this and we give that inhibitor at the same time, all of the animals live. So we're actually very excited about this idea, right? This is not a medicine. We have to give a lot of it. We have to give it simultaneously with the bacteria. It's not potent. It doesn't go, if I gave you this, you'd have to take a pill this big and it would kill you, right? And so anyway, we have to actually, now that we have some proof for this idea, the goal for us is to actually make this molecule medicine-like, right? To make it much more potent, to make it selective, to make it go where we need it to go. We have done none of that, right? We wanted to prove the point first, and those are the next goals for my gang. And so that's where we are in this story. And so the immediate goals, I just told you, is to do medicinal chemistry. So chemists know how to make molecules medicine-like, right? And so those are the features we have to build in, starting with the lead molecule that I showed you. And then what we'd love to do, remember I told you that Pseudomonas, this sticks on these catheters and sticks on these intubation tubes and on surfaces. Beyond making a medicine, what we'd love to do, and we're working with engineers now, is to make coatings. Like if you could dangle anti-quorum sensing molecules off of catheters or intubation, or you could make like coatings for hospital sinks. Like maybe you could also make sort of applications that aren't just the medicine. And so we're working really hard now with these material scientists to actually make biofilm resistant materials. We think that might be kind of interesting. We do this, I showed you the Pseudomonas experiment. We do this with lots of different pathogens. So there's many other, you could think about these kinds of applications for other pathogens. And then the real thing is to actually go back to that little antibiotic talk, is to actually ask, does it take longer for bacteria to develop resistance? They will get resistant, like we know that. But do you get a longer shelf life out of such a medicine if you change behavior but you don't change growth, right? You're just making them not talk, not hear, and giving the immune system time to get rid of them. So we have to, we have, the idea I went through is an idea. We haven't done anything to test that. And then sort of the bigger goals, or the big summary of this talk, is what I hope you think now is that bacteria can talk to each other. They don't have voices. They use chemicals as their words. I hope you think that bacteria are multicellular, right? So these ancient creatures figured it out, right? They figured out how to do collective behaviors and to join in groups, and they get much more bang for their buck by acting as collectives. And so we think that this could have been one of the very first steps in the development of higher organisms, and that some of the principles are embedded in these ancient bacteria, and we can learn about them by studying them. With the two molecules I showed you, I would argue that bacteria can tell self from other. And again, that's what happens in your body. It's not like your heart cells get all mixed up with your liver cells, and that's because they use different chemicals to give them fates and to tell them what jobs to do. We think, again, that some of those principles can be discovered in bacteria. And then, of course, there's these sort of applied part to this, is if we could develop strategies to impede quorum sensing in harmful bacteria. And so I showed you one example of our efforts there. But then I want to go back to my second slide, which is to remind you that there's this microbiome. So there's this magical set of bacteria that are actually keeping you alive. And so maybe the way forward in medicine is to stop fighting against the bad bacteria. But if we could actually learn enough about this microbiome and what quorum sensing is doing there, maybe we could make pro-quorum sensing molecules to make these guys chit-chat better. I mean, they are there working to keep invaders out, right? And so maybe we could beef up that conversation, and that's the real way forward in medicine. So we're trying both of these strategies, right? And I don't know if either or which is going to work the best. And then finally, I'll end with a confession that we didn't think any of this up. So once we got this idea, oh, maybe we could interfere with these molecules, we and lots of other scientists said, these bacteria have a 4 billion year head start. Maybe we should just go, they're fighting, they're doing all this stuff. Maybe we should just go out and look at what they've done. And so sure enough, when you go out into natural settings of bacteria, there's guys that eat each other's autoinducers. There's one guy that's making those molecules with the ring, and the guy next to it has an enzyme that cuts the ring off the chain, and the poor guy is talking, and the guy next to him is making it mute. They make antagonists, they free ride, they cheat, they eavesdrop, right? They do all of these things. So those are clearly out there. And those guys have the sort of natural strategies that we find out there, the ones that exist, have won over 4 billion years. I mean, they work. And so the idea that I and a graduate student is going to make a better chemical is ridiculous, right? And especially if you saw my graduate, no, okay, don't. Anyway, so maybe we should just go out there and mine this natural world and bring those in as the building blocks, and then we can tinker with those to make the next medicine. And so we have a pony in that race. A lot of people do, and I think that is going to be a treasure trove. Okay, so that I am done, not really, because I have to show you the people that I'm bad talking about, even though they're my heroes. So I want to tell you that these are tonight's heroes, not me. I have the incredible privilege to work with this remarkably dynamic and intelligent and creative group. And so, I get to have the good luck to tell you this talk, but in fact, these are the people whose brains and hearts and hard work made up every one of the ideas that I told you, right? And so I hope that, especially for any of the students in the audience or young people, that came, right? This is how science gets done, right? It's this demographic of sort of 20 to 30-year-olds mostly that are actually changing the way we think about the natural world, right? And so I'm really proud and I'm really lucky to get to work with them. And I want to say again that I didn't have any of those ideas, but somebody in this picture did every one of those experiments. And so I'm really proud of that, and I want to thank Harvard and MCB and OEB, and the museum for having me here. It's a real delight, and thank you for coming.